Good morning, everyone. We are Group 3, and Franco and I will be discussing Exercise 14, which is an in silico analysis of our unknown DNA sequence. As you can see here in slide 2, here is the diagram of the summary of our unknown DNA sequence. So the specific regions of the sequence are represented with their assigned colors. So orange for the 5' untranslated region, red for the open reading frame, purple for the 3' untranslated region, and black, white, and yellow lines for the start codon, stop codon, and highlighted amino acids respectively. So the 5' UTR is the segment before the start codon of the ORF. So it is usually at the beginning of the sequence, while the 3' UTR is the segment after the stop codon. So a total of 111 base pairs were observed for the 3' UTR and 41 base pairs for the 5' UTR. However, for the last ORF that has only 27 base pairs, the segment is from 3' to 5' direction already, since there is a start codon immediately after a stop codon, as you can see here. The best ORF was actually identified to have a complete set of start and stop codons. The reading frame also follows the 5' to 3' direction and has the longest chain with a total length of 183 base pairs. So the ORFs combined was observed to have a total length of 298 base pairs. So it has the highest number of base pairs in the unknown sequence. So as you can see here, the highlighted amino acids right before the stop codons were identified. These are glutamic acid, threonine, and leucine. So moving on to the next slide, upon the blast search of the unknown sequence, the following species revealed sequences shown to be of highest in-person identity. The DNA sequences are retrieved from GenBank in NCBI, and the first sequence that we found is from Octodontigus, and the second one is from Octodontimus scleroides, and lastly, Spalacoppus cyanus. Okay, for slide 4, you can see here the frames acquired from the translation tool on the Expasy website. So these frames were generated using the DNA sequences from the previous slide. So an open reading frame, or ORF, as we stated earlier, is defined as a portion of a DNA sequence that does not include a stop codon. So the open reading frames are seen on this slide as the ones that are highlighted in red. So there were 6 frames generated from each of the DNA sequences on Expasy, but only the ones with the best open reading frames were selected. So the best ORF was determined based on the following criteria. First is that it should be the longest one, as you can see here. It should start with M or methionine. It should have a stop codon, and lastly, it should have 5' and 3' untranslated regions. So as you can see, our ORF checked all the boxes.
Once the best ORFs were selected, we clicked on the M in methionine, or the start codon. This gave us the FASTA format of the translated product of the nucleotide sequence. This shows us the order of amino acids in the polypeptide chain dictated by the triplet codons found in the sequence. The FASTA formats of the top 3 known translated products were inputted into Clustal Omega to perform a multiple sequence alignment, which yielded the graph seen here. The numerous asterisks shown on the bottom of the sequences show the homology of the 3 sequences with a given unknown sequence. The phylogenetic trees given by Clustal Omega also show the relationships between the 3 known sequences to the given unknown sequence, which supports their homology. Attached on this slide is the raw data acquired from the Expasy Protparum tool. The gene has a total of 61 amino acids with an atomic composition of carbon with 310, hydrogen with 475, nitrogen with 87, oxygen with 86, and sulfur with 5, with 963 total number of atoms. Leucine garnered the highest amount of amino acid composition, having 18% of the total. This amino acid stimulates protein synthesis in muscles, tissue generation, and metabolism. On another note, phenylalanine has the lowest amino acid composition with 1.6%. This amino acid is involved in the production of neurotransmitters, neuropinephrine, and dopamine. Furthermore, this gene has a molecular weight of 6,957.01. In terms of positively and negatively charged residues, the positively charged residues, arginine and lysine, have 4 and are located near the cytoplasmic end of hydrophobic segments in membrane proteins. While for negatively charged residues, aspartic acid and glutamic acid have 5, and are in transmembrane regions that have a non-trivial role in the biological context. With extinction coefficient, 15,595 pairs of cysteine residues form cysteines. On the other hand, 15,470 of cysteine residues are reduced. For the estimated half-life, the N-terminal is methionine or the initial amino acid. This gave 30 hours for mammalian reticulocytes in vitro, at least 20 hours in yeast in vivo, and at least 10 hours for E. coli in vivo. Lastly, the instability index estimates the stability of a protein. If the value is less than 40, the protein is in a stable state. The instability index of the gene is 67.29, indicating that the protein is unstable. However, the value 92.79 was displayed by the allopathic index, wherein values ranging from 66.5 to 84.33 are thermally stable. The grand average of hydropathicity, which indicates the solubility of proteins, is negative 0.048. A negative value indicates that the protein is soluble in water. Two models were generated from the protein sequence of the 5' to 3' frame 3 of the unknown sequence, which are bovine insulin and B28-ASP insulin, or insulin aspart. Bovine insulin has often been used as a growth supplement in culturing cells at a concentration ranging from 1 to 10 micrograms per milliliter of medium. The amino acid sequence of insulin is extremely well preserved. Unlike bovine insulin, human insulin is an essential hormone for controlling blood sugar and energy absorption, and is a chemical messenger that allows cells to absorb glucose. It is only 3 amino acid residues similar to human insulin, enough to be clinically effective on humans. B28 insulin aspart is a rapidly absorbable human insulin that is currently being developed to treat diabetes mellitus. Cardiovascular and standard tests for behavior, neurological, and organ-specific functions have also been carried out in various animal species to test possible side effects of the insulin aspart. So far, the tests were compared with those of a soluble human insulin preparation, and aside from the side effects resulting from hypoglycemia, the overall pharmacological profile of B28-ASP insulin is comparable to soluble human insulin.